countries where the rice germplasm is submitted and is being maintained here in the International Rice Genebank. So maybe you can see your countries here. So all these countries are signings of the treaty where we are allowing the sharing of this material. So if your this particular country will sign for their germplasm materials to be stored here, they're also considering that their germplasm materials would be shared worldwide to these different countries which sign to the party also. So as of now, we are nearly 120,000 accessions of rice. That would include already the Asian rice, the Oryza sativa, the African rice, the Oryza glaberrima, as well as the more than 20 different wild species, wild relatives of rice. So later we are going to go around the Genebank and see the facilities and activities that we are doing to maintain this very huge collection of rice germplasm. So let us go here and look at the drying room. Yep, so come here closer. So this area here is the packing room. So this is the relative humidity inside here is controlled. So basically these materials already are from the storage or going to be stored already. So they are prepared and packed here. So the moisture content is already about 6 to 7%, which is ideal for in-bank storage already. So that's why the relative humidity here is quite controlled.
What is the RH here?
Same around this one, around 15% relative humidity.
15%.
To 20. It's quite fluctuating here, but inside it's more strictly controlled because this facility here is the drying room. Also, this is a temporary holding room that we use. So basically, the conditions inside is 15 degrees Celsius and 15% relative humidity. So with that condition, we are able to dry the seeds to the recommended by FAO and IRRI recommended moisture content of 6 to 7% moisture content. So that's we're following that protocol for it's a Genebanking standard. It's basically for most seeds, especially orthodox seeds such as rice. So we'll have a look inside. Yeah, so yeah, so it's quite cool. So again, with these conditions, we are able to dry the seeds around the recommended moisture content for about 2 to 3 weeks. In 2 to 3 weeks time, so we are able to dry the seeds already. So freshly harvested seeds would come here.
So the 15% moisture.
Yeah,
to
from the field
about
to 7
No.
to 7%. Yeah, in that
by just this drying.
yeah,
Temperature.
this condition, this temperature and combination
Temperature.
of RH and
15
temperature.
degrees centigrade.
No fungus?
Yep.
I mean, no any
No,
damage of seed?
that's the that's why we are drying this the seeds as soon as possible as harvested to avoid the fungal growth. So far there is no there should be no fungal growth.
Immediately after harvesting, you are bringing it here.
Yeah, immediately from the field,
Field to-
the field moisture content. Once the seeds are ready for harvest, they're immediately threshed and then dried already here. So aside from the drying room, we also use desiccants for some materials because these rice materials are very diverse. The germplasm, some of them would not dry down as quickly as the others. Like in 3 weeks time, it's still way above the recommended. We use also the these silica gel desiccants to accommodate those samples. Yeah. So the main point there is dry the seeds as soon as they are harvested from the field, and the target moisture content is 6 to 7%, which is ideal for the Genebank storage.
So all the seeds will have a uniform moisture content
Yeah.
after the here.
Yeah.
So immediately when come, you check the moisture content and put inside, or
Yeah. And then for in a regular period, let's say 2 to 3 weeks, they are checked for the relative humidity or moisture content. And then the materials will be moved to the sorting room area, sorting area, which we are going to have a look.
I'm drying, so let's go out. Malamig na, dry pa.
Kung natin sa 14 degrees nga mamaya dun sa?
Dehydrate.
Nagtuturo ka mo first time dito.
Sorting area. So once the seeds have been dried already from the drying room, so they are being checked here also for good seeds. So they are only selecting the good quality seeds. So they remove the unfilled grains as well as the immature grains and diseased grains. So this is some sort of a quality control before they are placed into the Genebank. So to do that, they have the seed file. So this cabinet at the back is the seed file. So they have the original seeds with which are their basis for checking these seeds. So each accession is being double checked here using the seed file. So they are sure that these harvested and regenerated accessions from the field are still the same as the original seeds, the original material that was brought here. So yeah, so in this area, these are some of the, you can see some example of the diversity that we have. So this is just a small portion of that. So you see how varied, how diverse alright sa Taiba is. This is Philippine cultivated rice, so much diversity. Here is the wild rice species also. So we have the wild rice materials, especially maintained at a screen house, a quarantine area for this especially for these materials. Because we don't, much of these materials are weedy types, so we do not want them to be contaminating our fields. So they are only being maintained in the GRC screen house. Yeah. This is a Tita Paula. This is
Okay. Hi.
Senior Genebank manager. So usually she's the one who does the cross-checking of the seeds from the seed file and then this harvested accession. So she must approve first that these seeds are the right accessions, so, and then they could already proceed in the protocol. So yeah. So maybe we could move closer here to the next part. Let's go here. So once the seeds are ready for storage, so we have these types of packing material. So each accession would have one bulk of, I think this is 300 grams to 500 grams maybe of seeds per accession. So this would be in the active collection. So important to mention is that the Genebank has two types of storage: the active or the medium-term collection, and the base or the long-term collection. So this is active because this is where the circulation and distribution, sharing of materials goes on. So in the active collection, we have one bulk and two pre-packs. With one pre-pack, we have 15 to 20 grams of seeds. This one is the one that is being distributed to requesters. So, and then also for accession, we have 2 cans of base collection for the base collection. These are vacuum sealed, and basically this packaging is moisture proof, so we don't want the seeds to increase the moisture content while in storage, so we would be ensured that the seeds are dried to the recommended moisture content inside the storage. And aside from the active and base collection that we have here in the GRC, we also have a duplicate storage for backup storage. We have one in Fort Collins, National Center for Genetic Resources in USA, and also we have one sample for the Svalbard Global Seed Bowl. If you are familiar with the, they call it the Doomsday Seed Bowl, where much of this germplasm that is being conserved is submitted to them. So we also have our rice germplasm duplicated in those. So we are sure whatever happens to the germplasm that we are keeping here, we have the backup, 2 of them for safekeeping. Yep. So now we are going to look at first one of the storage room that we have, the active collection. So in this active collection, it's also called the medium-term collection. We are able to maintain the viability of the seeds for up to 20 years, 20 to 40 years, without drastically losing the viability. So we are regularly also checking the viability. So theoretically, with these conditions inside, we are about to see and observe, we are able to maintain the seeds viability for such time. So let us go inside. So pasok po tayo sa loob. Yeah. So the conditions inside is much lower. It's about 4 to 5 degrees Celsius inside. So for those who want to have those jackets, you can wear them. But yeah, the relative humidity inside is no longer controlled since we have the seeds in a moisture-proof materials. So yeah, let us have a look. So, this go inside.
So minus 20 degree.
No, it's 5 degrees Celsius.
Oh,
Yeah,
5
much
degree.
lower than the 50
Oh,
degree.
this is
This is the
active collection.
So, so all the 120 plus thousand accessions are being kept here. So as we have earlier seen in the display, we have per accession we have 1 bulk and 2 pre-packs. So these are being shared to others. So once the pre-packs have been distributed already, so we take another, we prepare another sample of 10 grams from this bulk. And then once this bulk is being exhausted already, the seeds are all regenerated and multiplied, so that we have always an acceptable amount being stored. Also, the seeds are being checked for seed viability every 5 years in this collection, and also for seed health testing, because once the seeds have low viability already, we regenerate them already. So this is how the seeds are replenished regularly. So questions maybe at this point for the, so take some pictures and then let's go out. Yeah. It's quite cool.
Take one picture. Take one picture.
Yeah.
Come. Yeah. Very
cool resistant.
Thank you. Your freezing resistant. One separate. If I want to get one Genebank, you want to buy. Very good.
Ay, wala. Pag-crisis ko na siya. Hindi na makapagay.
Close.
Bye!
Bye. See you for the next 5 years. 5 years to rejuvenate the skin.
We cannot go out once this is
Oh, if you,
it's a double lock system.
okay.
Where you have to close this first before you go up. So maybe a further information that additional information maybe worth mentioning is that this facility is a fortified structure, which is they said is stronger than the building itself. So it is it make it ready for some catastrophes like earthquakes
That's it.
and other environmental factors. So maybe they're saying that this building can go down, but germplasm stay inside. Yeah, but you don't want to go inside in case of earthquake. Yeah, and also the power the generator of this one is separate from the building also, and then there is a backup. There are two backups of power supply. So yeah, this storage basically is very hard and expensive to maintain.
You also have duplicate in the seed vault of South Korea.
We don't have I think we don't have the whole collection, but we have their materials, RDA, the Korea RDA. We have, I think they have, I'm not sure if we have some materials there, but what I know is that their collection is also here. Yeah.
Okay.
So let's move on. So that was the actual collection. We are going to look at now the base collection, the long-term storage. So if you're familiar with the Harrington's rule of thumb, so that's a basically basic principle that we follow. So it was made by Harrington in I think 1972, where he stated that for every 5 degrees Celsius decrease in temperature, you double the lifespan of the seed, at least orthodox seeds. We're talking about orthodox seeds, and also for every 5% decrease in moisture content. 1% decrease in moisture content. You also of the seed. You also double the lifespan of the seed. So the lower the temperature and the lower the moisture, you increase the lifespan of the potential longevity of the seed. So the 6 to 7% is already the optimum. So beyond the beyond the 6%, you go lower than that. There is no incremental increase or increment in the longevity. So we don't want to use more resources in maybe going down lower than that. So the 6 to 7% is already optimum. But the temperature is much easier to control. So in the base collection, we are theoretically able to maintain the viability of the seeds for 50 years and above. So 50 to 100 years maybe. But the conditions there inside, the temperature is much lower. So that's the negative 20 degrees Celsius inside. So the materials are in the vacuum sealed aluminum cans. So we could have a look at that. So, ito po tayo. Yeah, we only have one jacket, but maybe we could just be Oh no. You'll get the jackets there. Okay, anyway, let's just have a quick malamig na yan. Shuttle. same number of accessions inside the active collection is the same number of accession fields. So we are able to pack more because we only give around 15 to 20 grams per accession. So yeah, this is the same 120,000. So you need 20 degrees.
And it's meant without frost.
Yeah, but this is actually, I think, relatively dry roast. So sometimes we see some frosting inside the door because of opening and closing. So yeah. So then let's go out. Some of them are freezing already. So,
Ayun, push the exit.
freezing up? Yeah, you see there is it frosting already. Oh no.
You have a special vacuum sealing facility there?
I'm not sure.
I almost embrace Andy,
Vacuum sealer is to be packing there. That we use, but it's quite useful. So that's how we.
Are we complete?
Yeah, we should be using.
2, 4, okay.
Yeah. So.
Mamamatay sa lamig doon.
So, questions so far for the two storage facilities? No, if none, we could go to the seed testing lab. Question? Okay. So, let's go to the, some of you might want
First
to
you
take
pack
Pictures.
and then you put this bag in this medium.
No, these actually are files. These files. Ito. Yeah, those are for seed filing. And I think this one is incoming. Yeah, these are new material that they are going to process maybe for planting and multiplication purposes. So if you notice also the materials, every accession, every pack is barcoded. So we have a system that we have Unity Resources Information Management system, GRIMS, yeah, that we use to handle and which aid the Genebank manager as well as the people here in the day-to-day activities that they handle. So each time they are pulling out materials, they use the barcode, and each time they are putting materials inside, they also track them by barcode. So this is mainly for tracking the materials and also monitoring the weight, the number of cans, the number of packets. So it's a very useful system that we have. And also to monitor the number of accessions that are already inside, the number of countries, the requesters, like that. So it's a database, basically a database management system we have. So maybe we could go to the seed testing lab. So maybe question. Yeah. And submit also for signal testing. Yeah, then you I think they also do a second stage drying materials because with moving materials, so some moisture in here, so some secondary and further drying is needed. But basically, once the seeds are passed the signal testing and also the other levels, no, just a sample, and if you run the sample, then also the weight, there's I think a threshold for the weight. 500 grams. They should pass that. The initial quantity before you store it. Because if not, they need to multiply that first. So So now let's go to the chip testing lab.
Yeah.
50 years. Berapa? Then, still viable for 50 years.